So on Monday, we talked about chaperones, we talked about PDIs, and we talked about folding pathway. So we just have a little bit more to finish with this PowerPoint, and then we'll get on to today's PowerPoint. And I talked about this on Friday, so I won't, that's all I'll mention it again here because this is the actual slide. And it's with proteins folding, that is the misfolding of proteins and diseases. And right now, we know of at least 35 diseases in humans that are associated with proteins not folding correctly, and they deposit outside of the cell into these insoluble protein aggregates. And we call these protein aggregates amyloids. And the diseases that they cause are amyloidosis. The most famous one being Alzheimer's. I guess Huntington's and Parkinson's also famous, but Alzheimer's being one of the best studied one. And this is caused by a protein called amyloid B. And what is interesting about amyloid B, and let me close my windows. Everybody doesn't need to hear dog barking. There we go. So what's interesting about amyloid B or amyloid beta is that it has no known function. So we don't know what it does inside the body. And like I mentioned on Friday, what happens is that these plaques will build, and amyloid beta is just like a beta sheet. And so you can kind of see it down here, is that you have these plaques and it's just beta sheet on beta sheet on beta sheet on beta sheet on beta sheet, building and building, building to form this plaque deposit. This deposit progresses as you age, and if you have enough of it, you'll start to kill your neurons, right? And one of the treatments is just trying to stop the rate of amyloid beta being created, because the less of this you have being created, then the less plaques that you will have formed. Can you find out ahead of time if we have a chance to have Alzheimer's? Based on your family history and such, yes, you can know if you're at higher risk for Alzheimer's. It is genetically linked, I believe. So like if people in your family have had it, then you are at a higher risk to get it. Right now, there's no way to prevent it. We do not have a cure for Alzheimer's, unfortunately. So yeah, it's one of those things that people are researching how to prevent it. Right now, we don't. Maybe in 20 years, by the time we get older, there is, but yeah, as of right now, there's not. If you'd like to be involved in finding a way to cure Alzheimer's, maybe a career in research is lines up well for you. But anyways, so that's protein folding and diseases. That's amyloid beta. Now, there are other diseases that are caused by proteins. And what's scary about these diseases is that you can transmit them between each other, between different humans, different organisms. Alzheimer's, you can't really transmit Alzheimer's to someone else, but you can transmit diseases such as mad cow, kuru, and Creutzfeldt-Jakob disease. And these are caused by Prion rods, and they're called transmissible, spongiform, encephalopathies, something like that. TSES, there you go, TSES. And the way that these diseases work is that there's a protein called a prion. And this protein is just mainly made out of hydrophobic amino acids. And let's say that you ate a cow that had mad cow disease. So you would eat a part of that cow that has a lot of these prions, a lot of these hydrophobic amino acids. Well, what would happen when you eat these is that you can have aggregation happening inside your cells, because Water is going to try and push all these hydrophobic amino acids together. Aggregation cause cell death. Kuru is brain disease you get from eating other people. So if you needed another reason not to eat people, it's so you don't get kuru. And the scary thing is we create prions inside of ourselves. You will create prions. Nobody knows what they do, why we create them, but they're there. And if you already create prions, you are at a higher risk to get one of these diseases, because basically you need this aggregation to start. So you need some kind of foundation. You need a protein there to begin with. And let's say you ate that mad cow. If you already had a prion, and in humans it's scrapie prion, PRP, they cause an aggregation event to happen. What's cool though is that if you do not create this prion, and some people don't, you are immune to all prion diseases. So you can't get kuru, you can't get mad cow disease if your cells do not make prions to begin with. Because remember, this disease is just due to protein aggregation of hydrophobic residues that can kill off your cells. So any questions about our prion rod disease? Don't eat people, please. I don't want any of you to get kuru. All right. So I do have a bunch of questions here. And I'll just briefly go over what the main point is. All of these questions are getting at the fact that to get these prion diseases, these amyloid diseases, you need a starting point, right? So in A, if I give prions in their normal mice versus mice that were genetically predisposed to develop a disease like Alzheimer's, why do Alzheimer's mice get symptoms first? Is because they already create the proton or the proteins to create this aggregation. And that's basically what A, B, and C are getting at. To have these diseases, to have these amyloid plaques, to have these prion diseases, you really need something to start it. And that something usually comes from your own body. So you either create prions or you create a lot of these amyloid beta proteins to create Alzheimer's. So if you eat people and you don't get kuru, you're good. No, it just could be that those people didn't have a lot of prions. You know, a negative, how to put this? A negative result does not mean a positive result. Just because you don't get something does not mean that you have something, or I don't know if that makes sense. Anyways, the lack of evidence is something evidence. I don't know. I cannot remember the phrase, but no, negative result does not mean a positive result. What if you cook it? So even with mad cow disease, I think cooking it did not unfold the proteins enough, because I know that was a big problem in the early like 2000s, late 90s, where they had to kill like millions of cows in England because people were getting mad cow disease. So I think even cooking it at high temperature did not prevent those prions from unfolding. No. So you need to study these proteins more if they're not folding when cooking. I mean, if you have disulfide bonds that are holding your protein together, it's not going to unfold with heat only. Yeah, so like disulfide bonds won't break under heat. But that would be my guess. All right, so that's all for prion diseases. Just know, what do you need to know from? You need to know what a prion disease is. You need to know what an amyloid disease is, and you need to know like how they're started. That's the basic information I'm looking for here. Let me share today's PowerPoint. Do I have today's PowerPoint up yet? I don't, so let me pop that up. Okay. And I hope everybody saw the news today about the Nobel Prize winner in chemistry. Wasn't me. If I won the Nobel Prize in chemistry, do you think I would be right now, 2:00 p.m. giving a lecture? I would be flying over to Europe to collect my $1.2 million. That's where I would be. No, it wasn't me. I am not that smart. The people who won the Nobel Prize in chemistry were Emmanuelle Charpentier. She's probably French, so I'm probably butchering that name. And Jennifer Doudna. I'm probably still butchering that name. And the Nobel Prize in chemistry was for CRISPR. These are the two people who pioneered CRISPR. CRISPR, and we talked a little bit about it this semester already, is the gene editing tool. Originally, it is a bacterial defense against viruses. And what researchers have done is they use this defense mechanism to edit the genome inside of a living organism. So they won the Nobel Prize for that today. They get the split $1.2 million among each other. So if you want to make it rich in science, just make something so groundbreaking that, who is it? It's not Swedes. One of those European countries give you $1.2 million. All right, so that's what happened today. That's what's going on in the chemistry world. So it's actually biochemistry. It's funny. They should just rename the chemistry Nobel Prize to the biochemistry Nobel Prize, because like everything is biochemistry related at this juncture. Yeah, it must be nice to get $600,000 for doing your science, for sure. I mean, to be fair, they have created a technique that has completely revolutionized the field of cell molecular biology, right? It's like the dark ages, and now we have light with CRISPR-Cas9. So totally deserving, 100% deserving. Yes, if you revolutionize the sciences, if you could give me part of your prize money, that would be great. I think I make as much as somebody who manages a McDonald's last time I calculated it out. So anything extra would be fantastic. All right, moving on. So what we're talking about today is we're talking about myoglobin, hemoglobin, and binding curves. Those of you who like math, you're going to love this. Those of you who don't like math, it's not that hard. I'll say that. So first, let's look at our protein. What we're looking at here is myoglobin. Myoglobin's role inside of the cell is more or less delivering oxygen to the mitochondria. So it's inside the cell, and it'll take oxygen from the cell surface and give it to mitochondria, right? The most important part, and what we're actually looking at, this and this are the same image, is what's called the heme. The heme is a prosthetic group on myoglobin that will transfer our oxygen for us, right? So the way that heme works is that you have an iron molecule right here in the middle. So we have iron, and it is being coordinated by a bunch of amino acids. Now, two very important amino acids, histine. So we have a proximal histidine, which is binding the iron non-covalently. So this is a non-covalent interaction. And we have a distal histidine. Now, in between the iron and the distal histidine is oxygen. So that's where our oxygen is binding. Does myoglobin only have one oxygen binding site? Yes. Myoglobin can only bind one oxygen at a time. Now, just looking at this image, we see oxygen fits in this nice little pocket where we're binding iron and we're binding the distal histidine. What binds better is the molecule carbon monoxide. So carbon monoxide outcompetes oxygen. I forget the exact numbers, but it's something in the range of like carbon monoxide will bind like 2,000 times better to heme than oxygen does. And the thing about carbon monoxide is you can't smell it, you can't see it, but it'll suffocate you. And the way it suffocates you is that it will bind to your heme and never come off. Well, if that binds to your heme, oxygen can't bind to your heme. If oxygen's not binding to your heme, you can breathe as much oxygen that you want. It's not getting to your bloodstream. It's not getting to your cells. So PSA for the day, if you do not have a carbon monoxide detector, you should get one because you won't know it's there and you won't know you're suffocating. 200 times? Okay, so I was off by a factor, 200 times. Oxygen is binding through ionic interactions. Nitrogen monoxide, I don't know off the top of my head. I would think because I'm guessing nitrogen monoxide does not bind as well as carbon monoxide. Because the thing with carbon monoxide is that it's really polar. And so that negative oxygen fits next to the iron really well. So it kind of fits like this. Nitrogen monoxide is going to be less polar. So it's not going to bind as well as carbon monoxide. I don't know how it does it against oxygen, to be honest. If you've been exposed to carbon monoxide, does your myoglobin forever broken since it doesn't want to unbind? Basically, yeah. But you make new myoglobin and break down old myoglobin all the time. So if it gets like bound to carbon monoxide, you're going to recycle it. I thought the gas companies put in some chemical and make carbon monoxide have distinctive smell. They could have, but like if you're sitting in a garage with your car on, then I mean you don't know carbon monoxide's through there for sure. Yeah, if they have, I haven't read that, but I would still get a detector. Oh, that's for natural gas. Okay, H2S. That would make sense for natural gas because you definitely want to know if you have a leak of that. And I don't see why the gas companies would be transporting carbon monoxide. Yeah, I don't know. Anyways, that's your lesson in health and biochemistry. So let's take a look at our graph here. Let's take a look at this graph. What this graph is, it's binding of oxygen and myoglobin. Our y-axis is the fraction of myoglobin bound. So myoglobin bound. It ranges from 0 to 1. 0, no myoglobin's bound to oxygen. 1, all of our myoglobin's bound to oxygen. Our x-axis is pressure, oxygen pressure. Oxygen is a gas, so we measure concentration via pressure. So this is just concentration of O2 measured in pressure. What we see is that as we increase the amount of oxygen, the amount of myoglobin being bound also increases. Now, there's a specific number we care about when we look at binding curves, and it's called K, or it's called KD. K and KD mean the same thing. It is your dissociation constant. Your dissociation constant is what concentration does it take to be 50% bound? Right? What concentration does it take to be 50% bound? So the way to figure this out is you go to a fractional saturation of 0.5. You draw the line until you hit your curve. then you draw the line down. Whatever ligand concentration that is, that's your KD constant. For myoglobin, it's like 2.5 torr, and torr is a measure of pressure, right? And for these KDs, the lower the number, the tighter your ligand binds your protein, right? Low numbers equal very high affinity, right? And this idea of being 50% bound, we call that the P50. So if somebody says like the P50 of myoglobin, what they mean is what pressure of oxygen does it take to be 50% bound, right? And we also have an equation to discover that or calculate that. So this equation will tell us how much myoglobin is bound at any concentration of oxygen, as long as we know the KD or the K. So fractional saturation, so that's what Y means, or percent bound. Fractional saturation equals concentration of oxygen divided by K plus concentration of oxygen. So that's a way we can actually calculate it. And what you do to make this graph is that you calculate a bunch of data points with different pressures, and you make a graph like this. All right, so this slide, even though there's not a lot of writing on it, there's a lot of information on it. So any questions about the information presented on this slide? Yes, you are going to calculate stuff using that, and we'll get to that this lecture of how to use it, but yes. If you know K only, how do we find O2? Basically, that's an experiment. Like if you know KD already, you just do an experiment where like, okay, if I know KD, what is the binding at 20 torr? What is the binding at 30 torr? Do we need to memorize equations? You should know fractional saturation equals pressure divided by K plus pressure. You should know how to do that calculation. So is concentration O2, PO2? Yes. For gases, when we talk about concentration, we always talk about pressure. That's how you measure concentration of a gas. So if we need to solve this, the pressure of oxygen will be given N to K. So there's a couple ways to ask this. One, I could ask you for fraction saturation. I would have to give you pressure, and I would have to give you K. That's the most logical way I'm probably going to ask this. I could also ask you K, but there are other ways to find that than from an equation, but you can find that from the equation. But mainly I'm going to ask you for YO2 and give you a pressure and a K. But we'll see today like what kind of questions I'm going to ask you, because we're doing those questions today. All right. So question 1, make a graph from the table and estimate K. So the idea is you would read this in and make your own graph. What I actually did is I kind of made it for you. So I'm not going to ask you to graph anything in this class. I am going to ask you to read a graph. So I forgot the 0, 0 data point. Let me put that in. Okay, 0, 0. All right. Based, and let me actually connect the dots. Based on this graph that I made, who can tell me what the K of my enzyme is? What is KD for my enzyme? And so let me just say, we're going to be learning these equations for myoglobin. These equations work for any protein that binds a ligand. So these are universal for any protein. We're just going to learn them for myoglobin. 0.5, 0.7, 0.8, yeah, something like that. All right, so we go to our halfway point when we're half bound, 0.5. We go until we hit the line, and then we go down. So 0.6-ish, something like that. Like on the test, I would probably accept anything from like 0.5 to like maybe 0.7, 0.8. Yeah, I guess that's okay. It's stretching it a little bit. But that's how you read a graph and you get your K from it. 0.5 is best. I would probably say 0.6 is best when I'm looking at that graph. But yeah, if you said 0.5, you're close enough. Yep, so that's how we read a graph and get our K. I did have a second question. Let me go back. So in muscles, in active muscles, when you're using your muscles, the oxygen, pressure of oxygen at the cell surface may be 10 torr. While when you go to the mitochondria, it may drop to 1 torr. What I want you to do, this question is worded a little funky, so I'll explain it. Myoglobin has a K of 2.8 torr. I want you to tell me the fractional saturation at 10 torr. I want you to show me the fractional saturation at 1 torr. And then imagine that you had 100 myoglobins. Out of those 100, how many myoglobins actually bind oxygen at the cell surface and deliver it to the mitochondria? So just to reiterate, tell me fractional saturation at 10 torr, fractional saturation at 1 torr. Then tell me from that information, if I had 100 myoglobins, how many of those myoglobins deliver oxygen? Might want to use that equation. So I'll give everybody a few minutes to get some practice with that. So I have an answer of 0.78 and 0.26. Yeah, that's correct for 10 torr and 1 torr. So out of 100 myoglobin, how many successfully deliver their oxygen to the mitochondria? It is not 26%. So while you think over that, I'll just show you the math that was done in case you're not quite sure how those numbers of 0.78 and 0.26 were gotten. So surface is just YO2 equals pressure, so 10, divided by K, which is 2.8 plus 10. Simple calculation, 0.781. Mitochondria. So that means 78.1% of mitochondria have oxygen. The mitochondria, where pressure is 1 torr, we get 0.263, which means 26.3% of or sorry, not mitochondria, of myoglobin are bound to oxygen? It's not half of what you got. It's not 10%. It's not 13.32. Okay, so this question's a little odd. Let me erase some of that. Maybe if I draw a picture out, it'll make more sense. I have a cell. And I have 100 myoglobin. At the cell surface is 10 torr. Based on what we just said, how many of those myoglobin are going to pick up oxygen at the cell surface? How many at the cell surface will bind oxygen based on the calculation we just did? .78 or 78.1. Yeah, so you can't have 1/10 of a 1. But yeah, so like 78. Now I'm just going to leave the decimal there. Okay. So 78 of our myoglobin bind an oxygen at the cell surface. Let's say all 100 of these myoglobin now go to the mitochondria. Once we're at the mitochondria, how many myoglobin are bound to oxygen? How did you get 78.1? That's that calculation over there, where the cell surface is 10 torr. 10 divided by 2.8 plus 10. Yep. So once we're at the mitochondria, now only 20.3 mitochondria have oxygen bound. Therefore, how many mitochondria, not mitochondria, how many myoglobin delivered oxygen? Nope, 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 nope. It's 78.1 minus 20.3. That is correct, which is 51.8%, right? So imagine at the first stop, 78.1% pick up its package. So 78.1% of my workers pick up their package, which is oxygen. They go to deliver their package, oxygen, to mitochondria. And if I check at their workspace, now 20.3% of mitochondria still have their package. They're still holding it, which means mitochondria, when we're in muscles, about 50% of your mitochondria are able to get oxygen from the cell surface to the mitochondria. So really your myoglobin is only 50% effective. Every 1 out of 2 myoglobin you have in your muscles don't do their jobs. Only 1 out of 2 takes oxygen from the cell surface to the mitochondria. So not that efficient. Well, luckily in this class, the math isn't too hard. It does take some thought about what to do. The actual math itself, not too bad. So questions about that idea, how, 51.8% effective. Where did 20.3 come from? Oh, that should be 26. That's me forgetting to write a 6. That's 26.3. So 51.8% is the amount of oxygen getting delivered. 51.8% is the amount of mitochondria that delivered oxygen. So kind of, it's just the amount of mitochondria that do their job. 26.3 is the amount of mitochondria, or I keep getting my 2 words mixed up. It's the amount of myoglobin bound to oxygen at the mitochondria, right? If you're a mitochondria, your goal is to take oxygen from the cell surface and unbind it at the mitochondria. You want to take it from the cell surface and deliver it to the mitochondria. So 78.1% of myoglobin can take it from the cell surface. But if you get to the mitochondria, 26.3% are still bound, right? So that means 51.8% of myoglobin can take it from the cell surface and deliver it to the mitochondria. Yeah, so some you need to think outside the box of how to answer that question, and that's why I understand it's an odd question. So, but yeah, it's odd to even understand what I'm asking in that question. I understand that. YO2 means how much is still bond. Yes, Y is fractional saturation. That tells you what percentage of myoglobin is bound to oxygen. All right. It's saying what percentage of your protein is bound to your ligand. So 26.3% is still holding the package while the rest delivered. Yeah, so if you had 100 myoglobin and you go to the mitochondria, 26.3% of that 100 would still be hanging on to oxygen. They don't do their job because your job is to let go. Yeah, it's not how much, it's like how many, because it's really it would, I could ask how many molecules of oxygen are delivered, and that answer is 51.8 molecules on every 100 mitochondria on average deliver, every 100 myoglobin on average deliver 51.8 molecules of oxygen. Isn't it because of the pressure? As the pressure changes, the likelihood of binding changes. This is what I got. 78 hemes picked up oxygen. Of those 78 hemes to the mitochondria. So mitochondria can accept all the oxygen. It's really 78 hemes bind oxygen at the cell surface. When we go to the mitochondria, 26.3% of hemes are still bound. So how many let go of oxygen? That's where the 78.1 minus 26.3 is coming up from. Right? Another way to put it, you have 10 guys or 10 people at a register. Everyone takes a $1, or 8 of them take a $1 bill. They go 20 feet to their right. Now only 2 people have a $1 bill. How many dropped a $1 bill? That would be 6, right? If 8 people had a $1 bill to begin with, and if I go to some point, now only 2 people have a $1 bill, I lost $6. So 6 of them lost their dollar bill. That's the same idea here. All right. So that was a long explanation, but that's how you use YO2. That's what YO2 means. And now we see how kind of crap myoglobin is at its job. It's only 50% effective. So I guess that's a good indication of that the body's not perfect. Just imagine if your body could deliver 100% of the oxygen to your muscles, you could probably run so much farther. You could lift weights. You could do way more reps of weights, but we're only 50% effective. Yeah. So that's a limitation. Hemoglobin is worse. Hemoglobin's way worse inside the cell. Hemoglobin does something different. Yeah, if you try, hemoglobin would be like 5% or 10% delivered. So hemoglobin is garbage at this role. That's why we have myoglobin. Myoglobin does something different. Fun fact though, I think I might have said this in the video, but mammals that go underwater, right, like dolphins and whales, they have a lot more myoglobin. So whale's not a mammal. Why did I say whales? But like dolphins, they have a lot of myoglobin to help facilitate this. So their strategy is just have more myoglobin in your cell, you get more oxygen. Whales are mammals. Yeah, I thought so. I was second guessing myself. But yeah, that's how they stay underwater for so long. All right. So I just talked about math for like 20 minutes. Do you still remember what I said about myoglobin, the structure of myoglobin? I have a bunch of true and false questions to see if you remembered, because we don't need to just know math in this class. We also need to know biology and proteins because it's biochemistry. So let's see if you remember what I said about heme and what I said about myoglobin. So let's see if we can answer a bunch of true and false questions. Get your mind off of math for a second. Is there a possibility it could be not beneficial for them to run at 100%? It's possible that for some reason it would not be good if we had 100% oxygen, because, little known fact, oxygen's a poison. So when we breathe oxygen, we're actually breathing poison. Our body's just really good at handling oxygen, the poison. Like, oxygen is like super dangerous. Oxygen ruins DNA, ruins proteins all the time. That's why you need antioxidants. So it is possible, yes. The other explanation why they're not 100% efficient is that it delivers too much oxygen and your cells just get destroyed from oxygen. So yes, good point. Interesting. The first three questions are basically even, true and false. Interesting. Leave it to mammals to figure out how to live in dangerous oxygen gas. Leave it to like all animals, right? I mean, birds can do that. Dinosaurs did that. Leave it to eukaryota for figuring that out. All right. Let's end this and share it. So, 1, the distal histidine covalently bonds oxygen. That is false. So, when we are transporting oxygen, we do not want to form covalent bonds. Remember, the idea of myoglobin is to transport oxygen. If we form a covalent bond with oxygen, we will not let it go. So oxygen is held in place 100% by non-covalent interactions. So no covalent bond. So 1 is false. 2, the distal heme binds iron. The distal heme binds iron. That's false. That's the proximal heme. The distal heme is out there, E7. That the distal heme interacts with oxygen, right? So it's the proximal heme or histidine that binds iron. So 2 is false. So 1 false, 2 false. 3 true. So I drew that little picture. When we have heme, it will bind carbon monoxide in a straight line. So that was a very fast picture. So I understand if you didn't catch that, but yeah. So carbon monoxide binds in a straight line. That's one of the reasons why it binds stronger. So 3 is true. So FFT, fast Fourier transformation. So 4, the iron in heme binds the oxygen iron carbon monoxide. True. So FFTT, a molecule of carbon monoxide can bind to myoglobin at the same time as oxygen. False. They bind at the same site, so you can't have both bound. So your final answers are false, false, true, true, false. So a good little review there of our myoglobin structure. Non-covalent. Yes, they are interacting non-covalently. Remember, histidine is positively charged. So what will happen when you put it next to oxygen, even though oxygen is a neutral molecule, it still has a lot of electrons, so you can still interact with the electron cloud. So non-covalent interactions. So that was all myoglobin. Myoglobin's boring. Let's talk about hemoglobin. And hemoglobin is a tetramer. That means it's made out of 4 protein peptide chains, 4 peptide chains. And it's really a what I would call a dimer of dimers. What that means is that it's made out of 2 alpha chains that are identical and 2 beta chains that are identical. So it basically looks like alpha, beta, alpha, beta. That's basically what your hemoglobin looks like. Hemoglobin found in your red blood cell. And hemoglobin has 4 hemes. So hemoglobin has 4 hemes. Right? Now, there are big structural shifts that happen between oxygen binding hemoglobin, where if we just look at the central cavern of our hemoglobin, when oxygen is not bound, there's a huge gap there. When oxygen binds, that gap closes. What that tells us as biochemists is that when hemoglobin binds oxygen, it makes the whole protein change. So the whole protein reacts to oxygen binding. And when we look at the binding curve, so myoglobin's up here, hemoglobin is this S shape. We call that a sigmoidal binding curve. And what a sigmoidal binding curve tells you is that you have cooperative binding. That was a bad line. You have what's called cooperative binding. What cooperative binding means is that if you bind oxygen at one site, the other sites, their binding affinities change, right? So the binding of one ligand changes affinity for the binding of other ligands. Hemoglobin has what's called positive cooperativity. So when hemoglobin binds one molecule of oxygen, it wants to bind other molecules of oxygen more strongly. So the binding of one oxygen leads to an increase of binding for all binding sites, which there are 4 of. And if you ever wondered why we have blue blood and red blood, it is because of hemoglobin. The iron in all these heme groups absorb light. When there's no oxygen there, it absorbs light to make it look like blue. When there is oxygen there, it absorbs light to make it look like red. So this is what's giving our blood the color. Is that why we turn blue when we suffocate? Probably because your blood is becoming deoxygenated. You don't have oxygen in your blood. So I would guess that's why. Yeah. That would be my guess though. So we're not going to get to the math, unfortunately, because we're out of time. We'll get to the math on Friday where we have more charts. Any questions about what's presented on this slide though? It is. Okay. Makes sense, right? Because if you don't have oxygen, you're in the deoxy state of hemoglobin, which appears blue. Hemoglobin has 4 hemes, myoglobin has 1 heme. Yep, that's correct. Basically, hemoglobin looks like 4 myoglobins stuck together. That's basically what hemoglobin looks like. They look like identical when you look at their structures. And we'll find out on Friday, hemoglobin's really bad at delivering oxygen inside the cell, but it's really good at delivering oxygen from your lungs to the cell. So if it has 4 hemes, it can pick up 4 oxygens. That's correct. Only iron binds to oxygen. Iron and that histidine is what's binding that oxygen. They make an oxygen sandwich. So let me go back to our previous slide here. This is a heme that we're looking at. This is the important part that binds oxygen. When we look at the whole protein, that heme is right here, right? That's the heme group. The rest of what you're looking at is not heme. The rest of that protein is just supporting the heme, right? So the heme is the most important thing, but the rest of the protein helps dictate how strong this heme binds oxygen. What's in the center? Empty space. There's nothing in the center. So let me draw my picture again, right? So that, I don't know why I'm using my hands. I have a pen. So let me just erase that crap drawing, right? So you have 4 subunits. They more or less look like that. Every time you have a subunit crossover, you have protein-protein interactions. Proteins, this is how they're talking to each other. Like, let's say, and we're going to get into this, but I'll give you a preview. Let's say I bind oxygen here. When I bind oxygen, the whole conformation of this protein changes. So I have a conformational change. When that conformation changes, the other proteins that are next to me feel that. They feel my amino acids changing. And through those interactions, they want to bind O2 more. So their O2 binding affinities go up. And actually, it's a whole chain reaction, right? But in the center there, you have no interactions, really. It's just like empty space. But we'll get into all of this good stuff on Friday. All right, any last minute questions about what we covered today? Is there any case where there are more or less than 4 hemes? There are specific diseases in your hemes. There are a lot of diseases, but there are some diseases where you can't make hemes well. So it is possible that like you're making hemoglobins with like an incorrect number of hemes or something like that. So if you have that, you are in a disease state. Cystic fibrosis is not that. Cystic fibrosis is that it's a specific water channel, or it's like an ion channel, I think. It's like a chlorine ion channel that you have one mutation of an amino acid, and you are no longer able to pump out like chlorine ions correctly. And since just one amino acid mutation ruins that channel, causes mucus and bacteria to build up in your lungs. Sickle cell is not a heme. It has nothing to do with your heme. What sickle cell is that you have a single amino acid mutation, one amino acid mutation, where you go from like a lysine to a valine. And so now you have like a valine right here. So you have a hydrophobic amino acid that's poking out into space. And what happens is that water does not want to interact with hydrophobic residues. And so it'll start to push all your hemoglobins together to form these long rods. These long rods are what give your red blood cells the sickle shape. And that is what will get them stuck in your capillaries. Because your red blood cells are no longer flexible. Can you ask a question from study guide? Shoot, go ahead. Porphyris, what is that? Porphyris are rare inherited blood disorders. People with these disorders have problem making a substance called heme. Oh, there you go. Yep. So there you go. Did not know that. Did not know that term. You have a sample of protein alcohol dehydrogenase. You measure the concentration of this protein at 280 in a 1 cm cuvette. The extinction if you measure the absorbance at 0.79, if the extinction coefficient is 1.89320, what's the concentration? That's Beer's law. Beer's law is concentration equals absorbance, molecular coefficient, path length. So that's usually C. Yeah, A equals E. So I got it wrong. Absorbance equals ACL. There we go. So our absorbance is 0.79. Our extinction coefficient is 1.89320. Concentration we're trying to figure out, and there's 1 centimeter path length. So solve for C. 0.79 divided by 189,320. So that's going to be a tiny number. Calculator. 1.89320. Concentration is 4.17 times 10 to the minus 6 molar. Yep. So the 0.79 is your A. Your E is extinction coefficient. L is like always 1 in Beer's law. Like I've never seen it not be 1. And then C is concentration. Any other questions people want to ask while you have me here? If not, same old, same old. I will put up a homework, put up this video. If you have questions, let me know. While I remember, test. When is our next test date? That has to be coming up soon. Let me pull up the schedule. Okay, our test next week, Friday. Yeah, so next week, Friday is test 2. So I'll be sure to remind everybody of that on Friday. Okay, good to know. Well, that's all I have for you. So take care, everybody.